Welcome to SciAnts Streams. If you are interested in all things science in a fun, wholesome environment, you are in the right place. SciAnts is a unique husband and wife team of Belinda and Lita, who hold their PhDs in molecular and systems biology from Dartmouth. What can you find here? The SciAnts team is dedicated to making science more accessible to our entire community. They believe that it is the responsibility of scientists to engage with the general public, and Twitch is one of their media doing this. By showcasing their own research, highlighting new discoveries, answering viewer questions, and sharing their passion with you. We do a variety of interactive science streams, including microscopy of ancient amber with insects, 3D printing, art, science news and deep dives, the occasional game, and more. If you've ever wanted to have a fun space to nerd out, now is your chance. We stream daily science content on Twitch. Understanding not just what we know, but how and why we know it is the essence of scientific knowledge.
And we have live ants too. They do not eat any of the leaves, Kanara. They will see, you can see they're walking across the little bridge over to the side. They're chopping the leaves, and they feed that leaf to the fungus garden. And then the fungus garden is what they end up eating. So they're farming the fungus. But I don't think I've ever seen them actually eat any of the leaves. They'll chop it and return it back to the fungus to allow the fungus to grow. But they don't have the digestive capability of breaking down any leaves. They don't have any cellulase to break down the cellulose layer. Will these ants like the taste of penicillin? So Hugin, that's an interesting question. The fungus that they have inside their fungus garden actually releases antifungals and antimicrobial peptides. And you might be like, Blint, why does a fungus release an antifungus? That doesn't make any sense. So the antifungus that's released by the fungus is to kill other fungi that could maybe compete with it. So it's not toxic to the fungus that they're eating and they're farming, but if any other fungus tried to colonize, it would kill off that competition. As Alex says, yeah, it would kill off that competition. So I don't think, Hugin, that they eat penicillin, nor they taste like it, but there might be some undertones of an antifungal in the fungus. Hugin, I've actually never tasted the fungus. I have no idea what it tastes like. Nor have I tasted penicillin, so I don't know if the ants would necessarily taste like penicillin or not. Bot says, what proteins, minerals, and vitamins did they get from the fungus? So Bots, great question. Unfortunately, I am not aware of any study actively looking at what the composition is of the fungus. So what all is it comprised of, and then looking at the gut of the ants. There's been some studies to look at the gut microbiota of the ants, meaning what are the microorganisms that live in the gut of the animal and that help them break down the fungus. And they found that there are important bacteria that live in their gut. But honestly, given this is their primary diet, so they must get everything that they need from the fungus. Thank you, Alex. Yeah, this link that Alex just put in talks about how fungus grows specialized organs, which we call ganglidia. Inside the ganglidia are fats and proteins, which are nutritious for the ants. The ants eat these. Let's see. Yeah, and here's a good look. Hold on, guys. At what the fungus looks like. We also have a 3D model of it, too. And so there's the different component. There's subcomponents that they're eating that have the fats. I've always identified it based on color. So things that are dark gray, those are the new growths. Those they don't eat. The orangey ones that you see in this image, that's dying fungus. They don't eat that as well. They eat the middle row, the white in color, and that's where the fruiting bodies are that they end up eating down. Also, I've read that they eat those fruiting bodies down to prevent the fungus from sporing. That's a little bit of an extra bit there as well. Webbed lattice is by design or accident. So it is design, Bots. So that lattice, they put the leaves in that orientation to make the fungus grow in a particular way. And actually different nests will have different composition of these tunnels. There are some that like to have bigger tunnels. There's some that like to have smaller tunnels. And even within the same species, you're going to see that level of variation. And why that is, people speculate that each colony has its own little personality, which I think is true. I don't know what the genetic basis of that is, but even the same species that, like, of ant that were identified, like, they're in the same mating flight and they're in the same island, will have such differences that, like that we just call it personality differences of the colony. And it must be some genetic relatedness, but I don't know, like, why the same species would have such vastly different looking structures. But yeah, they actually, all the bots, the brood, the embryos, the developing eggs, larvae, pupae, are also packed away in those tunnels. So their underground chambers are circular, and they're filled with this fungus. And in that fungus, there's soldiers, there's nurses, there's cutters, there's gardeners, there's all the cast doing their work. Even the queen is somewhere in this pile of fungus. And they don't have separate rooms without fungus. Every room, independent of, like, the connecting tunnels, are filled with fungus, and that's where everyone hangs out. The connecting tunnels are just where they walk around at to get from new places. And the other thing they do, in fact, do too, is they'll have a trash room, so a dedicated trash room at the lowest chamber of the colony. And that is to, if they had put it higher and there's a collapse, then the trash would land on the fungus and infect the fungus and kill it. So that's why they do it like that. Is the mycobiota a huge group to study too? So all the gut cells, Chris, there is a ton of additional, like, a whole gut microbiome that is influencing not only the behavior but also the, like, what they digest, what they eat. And so that is studyable, but there's a lot of ways to study it so you can get all the different answers. You can do proteomics, so decide what the proteins are that comprise the microbiome. There's also genetic analysis, or RNA sequencing, but you have to make sure you're parsing out what is from ant and what is, like, the gut microbiota, like what are the bacteria that are living on there. And that is a technically challenging series of experiments that's also expensive. The fungus becomes the nest. So inside, so everything's underground, and inside of the nest you just see fungus. So let's actually so Chris, are you part of the Discord? I have my own personal photos too. Let me actually see if I have any of if I've uploaded any of those photos. It's always easier to use those. So this one is a good look at them adding the leaves and actually designing the chambers. So all of this up here, you see the green? That's freshly added leaves. Oh, that's right. Yes, Chris, thank you. There is, Chris, lower down under, like, Belinda and Lita's projects, there is an ant photos channel, and I'll put in some extra photos. I've been neglecting it. I usually try to put up one a day, but these photos are here that I've taken. And so this one, you can see as they're adding the leaves, and so that's that green there is the new additions, and you can see the hyphae are growing around and enveloping the leaf in the shot. And then I have one earlier, so I removed some of the fungus from the pile so you can actually see a little bit. Yeah, here we go. This is our little colony. This is many months ago, but you can see the queen is right here. She's the big one. And then all those little shapes that we're seeing are the leaves being added. So they add the leaves to that shape. So imagine this box that they're in is an underground chamber. And they fill up that chamber with fungus. Once that chamber is full, well, and during too, they're off digging another chamber that they're then also gonna fill with fungus. And they will keep filling rooms with fungus as long as the fungus is growing. And so you can think of it like a very long-term project that they have. They're going to be adding them 20 plus years even. Some of the queens can live up to 30, 35 years. And so the longest one was like, I don't know, it was like 1,000 meters or something like that, even larger and deep underground, so that people, like the height of a person, it was actually absolutely nuts. Yeah, they get huge, Chris. I'll find an image for you in a sec. Dapper Demon, welcome in. How you doing, Dapper? It's good to see you. Chris, let me find you an image. And then I know there was other questions. I'll get to other questions in just 1 second. This is the most striking one. This image is in one of my favorite books called The Leafcutters by E.O. Wilson. And so there you can see like humans as references for how big that colony became. And each one of, if we zoom in, there's little balls here. Here we go. These little balls are all chambers that are full of fungus, which is pretty remarkable. Those would easily be the equivalent to surpass a projected size. Yep. Yeah. Oh, yeah, Chris. It totally looks like a sci-fi colony. Doing all right, Dapper. How are you, Dapper? All right. I'm going to scroll up on some of the questions. Bot says, have there ever been any experiments to determine if the queens have some sort of anatomical difference in their brains beyond just overall anatomical... So, okay. So, bots, what... Actually, this is experiments that I've done. And other... So, So, I've done this experiment in leafcutter ants, but the experiment's also been done in other species of ants. So, what was identified was that there were different cell populations. So, anatomically, bots, the only difference is size. And maybe, depending on what cast you compare it to, like the optic lobe might have more cells because they need to be more visual because they need to be able to fly more. Because the nurses, for example, have a fairly small optic lobe, but they never will fly, and they're also always underground. Versus the soldiers have a larger optic lobe, kind of like this model here. So they have these long stretches of like neuronal tissue from the eye that terminate at the retina, versus like a nurse will have something like this where you have like a bulb of the optic lobe and not this long stretch that ends at the retina. But the big thing, bots, is doing single cell RNA sequencing. So identifying on the single cell level what are the genetic differences of those cells and what are the cell populations. And that shows that there are different cell populations in the queen brain that's different from the worker brains. So then it's just beyond just size. Genetically, they're very different. I guess I'm getting at is, they are chemically determined to be a specific type of cast of ant. How do they know what they are? Oh, bots. Bots, that is the bajillion-dollar question. So what Bots is asking is, how does an ant cast know what it's going to be? When they're laid, Bots, as eggs, they are all genetically identical. Okay? It is not determined at the point of being laid. Every egg at that stage can become whatever, like any of the casts. When they hatch into larvae, they start being fed by the nurses. They do not eat the food by themselves. They need to be fed. So, which makes the experiments, like, if you're trying to genetically modify a larva, you have to have it with adults to take care of it, which is a little bit difficult then from an experiment question, because it could be hard to then ID, like, if they're hatching, right, who was the adult and whatever, because they also are kept clean. The feeding, the ones that feed, the nurses that are feeding the larvae, they regurgitate the food into the larva's mouth. When they're regurgitating the food, we think they're also regurgitating compounds that are epigenetic, meaning that they modify the genome. They don't cause mutation. They simply close or open certain genes and sites of genes. So the DNA could be scrunched up, so that none of those genes that are scrunched up are going to be turned on, or it could be opened up, so that those genes can be turned on. And we think it's that that regulates this on-off state that gives them to be what size they are. Now, it's not just that much of feeding. We think there's also genes that are turned on that do the same thing inside the larva. But that initial spark, we think, is from the feeding. We don't know what that compound is, but there is a molecule called CoRest that does the same thing as this feeding idea. And that is expressed in the brains of the adults until about they're 5 days old. And if you change the levels of those regulators up through day 5, you can permanently change their behavior. So you can make a soldier behave like a nurse if you turn off that molecule. Or if a nurse, if you turn it up, then you can make it behave like a soldier and not do nursing behavior. It is big game. It's a fascinating field of study. And why ants? It's because they are genetically identical, but you're getting wildly different results. And so it's all how genes are regulated, which is a very complex question to ask. Like, how does this work? I don't know. And so it's the ideal organism to ask that question on.
Thanks for watching SciAnts Media, and be sure to hit those like and subscribe buttons to help our channel grow and support our science outreach and education goals. We can be found on Twitch most days of the week, and be sure to join us here for our next video. Thanks again, and stay curious, everyone.